Everyone, well, I believe you can all see. Okay, hi guys, I'm Julia. Just as Peter mentioned, I'm in the control lab and I've been at Queens for just over a year now and I'm really enjoying it. It's been great. So I'm going to first start with this working. A quick refresher on what is multiplex immunofluorescence. Let me review what's everything that's happening up top. Okay.
Is your volume up?
Yeah, it is.
Any complaints about the volume?
Okay, so here's a quick refresher on what is multiplex immunofluorescence and what it's used for. So multiplex immunofluorescence is a tool that has come to the forefront of cancer research over the past 10 years because it's something that we are able to use to study the tumor immune microenvironment, something that Peter just alluded to. It's very useful because we're able to maintain the spatial relationships of cells in situ within the tumor and look at their cell-cell interactions to understand how different communities of cells might correlate to outcome and thus help us determine who before treatment will respond to the treatment once they go on it. With multiplex immunofluorescence, we can add up to about 9 markers on one histological slide at once. And what you're looking at here is one of the panels that we have developed with our sister lab at Johns Hopkins, and it's called the Axis panel. I'm going to walk through each marker now in this photo and keep them in mind because you will see them over and over again throughout my presentation. So in the top here, you're looking at FOXP3. FOXP3 is your regulatory T cell. It's in pink and it's littered throughout the image here. Again, LAG3 is a known immune checkpoint inhibitor. PD1, PDL1, these are very common immunotherapy targets. I'm not going to dive into that. CD8 is your cytotoxic T cell lineage marker. CD163 at the bottom here is your macrophage lineage marker. And then as you can tell from this panel, all of these markers are very immune specific. So the purpose of this panel is to understand the immune component of the tumor immune microenvironment. That's what it was designed for. A1, A3 down here is a pan-cytokeratin marker. It is what marks the tumor. Okay, so over the next 40 minutes, I will be giving you guys a quick overview of the multiplex immunofluorescence workflow from tissue sectioning to phenotyping while focusing mainly on cell segmentation, which happens after imaging. And then I will move on to discuss our new addition of a robust membrane stain to all of our panels to generate accurate single cell data from our images. And then I'll move on to discuss the troubleshooting I have done to optimize that membrane stain within multiplex panels of markers. Okay, so to start, the overview of multiplex immunofluorescence. So we start with a formalin-fixed paraffin-embedded tissue. So this can be a resection or a biopsy that is thinly sliced and placed onto a histological slide. It is then processed, so all the wax is removed from the tissue and then stained sequentially with our TSA Opal technology on our automated stainer. Once that is cover slipped and dried, it is then stained by our multispectral scanner. And then those images are uploaded onto our image analysis software, which starts with cell segmentation to start processing the images. Cell segmentation is where the software will go in and try and trace the membrane of every cell to then label the cell as one. And then we move on to phenotyping, and phenotyping is where the software will go in and label all the markers that either fall on that border or within those borders to then phenotype the cell. So as you can imagine, one of the bigger questions and something that Peter just alluded to is how is accurate quantitative single cell data pulled from our multiplex images? How are we able to get single cell data from a photo? Well, that starts with 2 key things. The first of which is careful multiplex immunofluorescence panel validation. This has been discussed already in many of our seminars, specifically by Leanne. She has explained in the past how we go 1 marker at a time to validate them and then put them all together and make sure that each marker maintains its true signal when in multiplex. And then what happens next is once we go into image analysis, we then upload these images into our software, and then we tell the software which markers within the panel are on a membrane. So in the case of this image here, if you look at the green, you'll see the green is on the membrane here. And then if we look for some purple, pink, and blue, those are also on the membrane. We then tell the software that those are membrane markers. The software then goes and finds those markers and traces the cell, labeling it as such. This is a really important step within the software because you need accurate cell segmentation in order to get accurate single cell phenotyping. And we have run into issues with this, which I'm going to walk through now. So, like I just said, the bedrock of accurate data for multiplex immunofluorescence is cell segmentation. And some of the issues that we are seeing with cell segmentation currently are under segmentation, where we end up having two cells labeled as one, and this is a problem for phenotyping, and also just for general cell counts. We'll get an incorrect counting of how many cells are in our image. The next step, we have over segmentation, where in this case here, the DAPI, which seems the nucleus, is very dark in the center. This is a large tumor cell, and the nucleus is quite large. The algorithm doesn't quite understand that this is just one cell, and it believes that the space in the middle is dead space and draws a line through it, making it two cells. This is also a problem. Last but not least, we have missegmentation. Now, in this case here, what happens is the algorithm is starting with the nucleus and trying to build out to find the outline that we told it as such, and it stops short. It doesn't quite reach the membrane it's supposed to get to. And this is a problem because then it will just label all the markers that fall within or on this border, missing all the markers on the outside here, misphenotyping the cell. So these seem like relatively small errors when we look at them, but when we think about the amount of data that we are working with within our projects, it gets very big very quickly. So with just one resection, so just one slide, we have about 427,000 cells. And then when we think about that in the next layer up, we tend to stain about 3 to 5 serial sections from each patient with different panels that we have developed. That gets us to about 2 million cells. And then if we go the next step up, we stain many, many patients, about 20 to 25 in a general cohort. That gets us to about 40 million cells. So when you have small systematic errors like that, what does that mean when it comes to bulk cell counts? Well, our sister lab down at Johns Hopkins, the TAB lab, they did look into this. And what they looked at was to try and understand how the current gold standard of cell segmentation is missegmenting a lot of the cells within multiplex immunofluorescence. And what they found is the current standard, which is the single pass approach, where you use one cell segmentation algorithm to segment all the cells within the tissue, we end up getting an over segmentation of the larger tumor cells. And the reason for this is that these single pass approaches are very, very good at getting the small leukocyte-like cells within our tumors. They, however, fail at getting the larger tumor cells, and this would make sense as we start seeing very leukocyte small segmentation within this approach. What the TAB lab then went and did is they developed a new approach where they used 2 different algorithms to analyze their images. They used one algorithm to look specifically at the small cells, and then another separate algorithm to look at the large tumor cells. And they were able to remove that over segmentation that we were seeing with the original single pass approach. And then they did a more, and then they looked at the bulk cell counts. And what they found is we looked specifically at the tumor cells here and also at CD163, which are your macrophages. There's a reduction in about 25% from the original approach where we had too many cells to the new approach. And this is major. If we go back to what I was talking about the slide before, if we have about 40 million patients and there's about a 25% difference, that can mean that either 10 million of those are wrong, or we actually end up counting 50 million cells and about 10 million of which are wrong. That's a problem. However, we know it's a problem. So how are we currently catching this bad cell segmentation? Well, bad cell segmentation is currently being caught by human annotation and validation. Unfortunately, someone has to go through all of our images, look and see how the algorithm is performing, and then recircle and retrace the cells that are wrong. And that is time consuming and not useful for a clinical approach, which is the end goal of our technology. So how can we improve cell segmentation? And this is where my project comes in. So my project is focused on optimizing membrane staining. So currently, any marker that identifies a membranous a membranous protein, let me repeat that. Any marker that is currently on the membrane and will be stained by our panels can be used to train the cell segmentation software that we have. So while our panels currently have markers like PDL1 and PD1, which will get the cell membranes, there are many cells within our tissues that do not express those markers. So how do we pick them up? And that's where my goal comes in. My goal is to develop a generalized membrane stain that will stain all the cells not already outlined by markers currently on a panel. And we call this generalized stain, from now on, I'll be calling it the PanMembrane stain. PanMembrane as in it will get all the membranes. There are three main aims to my project. The first aim is to improve the input image for cell segmentation by removing background staining in the current PanMembrane stain. So if we go back about a year, when I inherited this project, the original the original cocktail concentration that I got was creating images that had a ton of background. We weren't getting accurate cell segmentation. There was a lot of missegmentation happening within those images. My second aim then will be to validate the new membrane stain's performance in multiplex images. And then my third aim is to hopefully identify a better segmentation software than the current platform that we are using. Okay, so this here is a gorgeous multiplex image of the axis panel. This is the same panel that I showed you guys at the very beginning of my presentation. In the dark blue here, which is scattered everywhere, this is DAPI. Those are your nuclei. That's a single cell. And then if you look at the green here, this is PDL1, which is outlining a bunch of alveolar macrophages. If we look for the orange, that is our pan cytokeratin marker. Those are all of your tumor beds throughout the image here. If we look for the red, these little red circles here, nicely outlining a bunch of cells, those are your CD8 cells, your cytotoxic T cells. What else is there I can point out? And then in here, these are leukocytes, which is a mix of the red and the pink, which is your macrophages. But if we look over here, which I'm going to focus on for now, we are actually missing some staining. I'm going to zoom in on that. We look down that bottom corner there, we see that there are a lot of the dark DAPI nuclei, but there is nothing tracing them. There's no staining. These cells do not have a membrane marker on them. They will not be picked up by our image analysis software. So just to highlight them there. Does everyone see what I'm talking about? They're missing? Lovely. So my job was to build a stain that will pick up on those. Now what you're looking at is the exact same image, but in this case, I've added on an 8th marker, and that marker is PanMembrane, and it's in white. And if we go back down to the bottom corner down there, we see is that missing cell is now beautifully outlined here and again here. So what is in the PanMembrane stain? How was I able to create a cocktail that will get all of these cells to be on current packaging? Well, the PanMembrane stain is a cocktail of 3 different proteins markers. It is 3 different antibodies to 3 different proteins. And those 3 proteins are CD45, CD44, and a sodium potassium ATPase. And I'll walk you guys through each one individually now. So the sodium potassium ATPase is the workhorse of my cocktail. Sodium potassium ATPase is found on all plasma membranes of almost all animal cells. However, it is upregulated on tumor cells. It is made up of 2 subunits, an alpha pump and a beta scaffold. However, not as relevant to me, but that my epitope that my antibody binds to is on the inside on the scaffold, on the pump. The antibody type that I'm working with, as is for all the antibodies we'll be talking about today, is an IgG anti-human monoclonal antibody. So ATPase is the workhorse for my tumor cells. CD45 is the workhorse for all my leukocytes. This is a leukocyte common antigen. It's a transmembrane glycoprotein, and it's found on most nucleated cells of hematopoietic origin. My antibody is able to recognize all 5 isoforms. It is once again an IgG anti-human monoclonal antibody, and it comes from a mouse. If you notice on the last slide, the ATPase came from a rabbit. And then last but not least is CD44. CD44 is another leukocyte-specific marker that I'm using in my cocktail. And the reason I'm using it in addition to CD45 is because when I look at the staining of CD45 alone with ATPase, I was getting very inconsistent staining where it was not even across all the tissues and all the patients I was working with. And I wanted to even that out. And the way that I did that was by adding in CD44 and it evened out the staining across all tissues. Again, another IgG anti-human monoclonal antibody. Okay, so now I'm going to talk to you guys about my first aim of my project, which was to improve the input image for cell segmentation by removing the background staining. So when I inherited this project, the PanMembrane stain looked like this. If we take a second and just kind of take in the image, what we'll notice is that there's a lot of background staining within the cytoplasm of the cells. And there's not a very high contrast of the membrane to the cytoplasm. Looking specifically here, there's really, you can't tell largely the difference between what is membrane and what is background. And same thing up here. If we think back to the issues I presented at the beginning of this presentation, where we have missegmentation, oversegmentation, undersegmentation, an image like this would be very difficult for an algorithm to work with. It will get most of these cells wrong. So how was I able to improve the image? Well, in our lab, we work with a technology called TSA Opal technology. It is a type of indirect It is a type of indirect immunofluorescent labeling, in which that means that we have a primary monoclonal antibody, which is bound by a secondary antibody, which then catalyzes a reaction of an inactive fluorophore to become an active fluorophore. And this inactive fluorophore here becomes a free radical and then binds to the residual tyrosine residues around the epitope of interest, creating an amplification of signal. So in a more direct type of immunofluorescent labeling, you'll find that it's one fluorescent label per epitope. In the case of our technology, we get a lot of signal for one epitope. And that's great for markers of interest, such as PD-1, which is lowly expressed. But when we think about the markers that are in my cocktail, ATPase, CD45, they're everywhere. They're highly expressed. I don't necessarily need this high amplification of signal for my marker. So what I did to address the high amount of background that I was seeing in my initial images is I reduced down the primary monoclonal antibody. And by doing that, I was able to remove most of the background staining. If we look at it again here, we'll see I get much crisper outlining of the larger cells, just largely absent over here. And yeah, it worked. It was exciting. And so to give you guys some numbers of what I was working with, for CD45 and CD44, I was at a 1 to 200 to a 1 to 100 concentration. I brought those both down significantly to 1 to 24,000 and 1 to 1,600. Then with ATPase, the workhorse of my cocktail, I had to dilute that down quite a bit. I was getting a lot of background staining from this specific marker. I brought it down to 1 to 80,000. And as I alluded to previously, my three markers do not all come from the same primary species. They come from mouse and from rabbit. And so I have a cocktail of secondary antibodies as well. It's a cocktail of a cocktail at 25% anti-mouse and 25% anti-rabbit. And the last thing I did when optimizing this stain is I looked at the 3 different lung cancers that my lab is currently working with, and I found that it worked across all 3, which was very exciting. Over here, we have adenocarcinoma, lung squamous cell carcinoma, and then mesothelioma, which is that was exciting. Yeah. Moving on to my next aim of my project was to incorporate that newly optimized dilute stain into a full panel of multiplex markers to see how that would work and hopefully remove the background staining that we were seeing with the initial PanMembrane stain. So the easy part was over. Diluting a primary was actually the easiest part of my project, unsurprisingly, because when I threw it back into a full multiplex, the cytoplasmic staining came right back. Unfortunately, what we're looking at here is this is my stain by itself. There's no other markers on the tissue. When I put all other 8 markers on the tissue, suddenly I have this really dark, arguably worse cytoplasmic staining than what I started with. So instead of freaking out, what I did is I went through each of the other 8 markers on the slide and looked for what I thought might be the problem. And I immediately fell upon our pan-cytokeratin marker. And the reason I fell upon this marker is because when you look at the image over here, I could quickly pick up that all my leukocytes were staining appropriately. This is what I was used to seeing in my previous images. But all of the tumor in here, this was not normal. This was unexpected. When I look over here, it's an exact match. So everything that we're looking at that's gray and gross in here matches what's happening over here. My next question was, why is that happening? Well, the first thought I had was perhaps there is some spectral overlap, and the signal that's coming from this image here is bleeding through into this image here. And so I went to look at that. And what you're looking at here really quick is all 7 of our markers here. They're spectral bands and their corresponding overlap. And what you'll find is there is very minimal overlap between most of our emission and excitation spectra, but that there is one here that's quite large. So let's go back to my problem. Is there bleed through happening? My marker, the PanMembrane marker, works in the channel 780. So it's the last channel in this row. And there's only one other channel that can bleed into it, and that's channel 690. So unfortunately for me though, AE1, AE3 or the pan-cytokeratin marker is not in channel 690, it's in channel 620. This is not a bleed through problem. There's something else going on. So I thought about it some more, and I was wondering, the pan-cytokeratin marker goes on before the pan-membrane stain. And with that in mind, I was wondering that perhaps, although this is not ideal and not how our technology normally works, there might be residual primary antibody left on the tissue by the time we stain it with our pan-membrane stain. Because like I said before, I have a cocktail of secondaries within my pan-membrane stain that are both anti-rabbit and anti-mouse. So I'm going to quickly walk you guys through why this shouldn't happen. So the way that our technology has been optimized to ensure that we're able to layer on many, many different colors onto one tissue without getting this cross-reactivity is through this reiterative stripping step at the end here. So we'll start with tissue prep, we'll retrieve the epitope retrieval, and then we'll stain with our first marker. And like I said before, the primary, the secondary will go on, and then there will be a reaction, and then the fluorescent markers will covalently bind to the tissue. Now, this covalent binding ensures that when we go through the next step of antibody retrieval, we don't lose that fluorescent signal, but we do lose the primary and secondary antibodies. And I'm going to quickly show you guys that in a better way here. So we have our initial staining, it's all done. We have a nice amplified signal. We then wash off the residual fluorescent molecules. We then heat it up and apply a solution at a pH of 9 or a pH of 6 to really shake off those primary and secondary antibodies, leaving behind nice fresh tissue for us to go again with a new marker. Okay, so back to my problem of weird staining. When I look at the panel's characteristics, like I said before, the pan-cytokeratin AE1, AE3 goes on right before my pan-membrane stain in position 8. And I have overlapping primary species. So my CD44 and CD45 are the same as AE1, AE3. And so my question was, are there residual AE1, AE3 primary antibodies after stripping? And are these being detected with the anti-mouse secondary used for CD44 and 45? And so I did an experiment to look at this, and I did a dropout experiment where I dropped out the secondary antibodies in my pan-membrane stain. So let's start up here in the top left corner. I'm going to quickly orient you guys. All these corner boxes here that have a little bit of orange stain in them, that means that these images also have AE1, AE3 pan-cytokeratin marker in them as well. In the corner up here, it's just my pan-membrane stain that is not there. So what's happening up here is we're getting that gross staining as I saw when I initially did the first multiplex. And then we look down here at this, in this image here, I've dropped out the anti-rabbit secondary, which means I'm not getting any of the ATPAs. None of the tumor cells should be stained, except I am getting a lot of tumor cells. I'm getting very, very dark cytoplasmic staining. And then when I look over here, where I do keep the rabbit antibody, but I drop out those mouse secondaries that were supposed to stain my leukocytes, my CD44, my 45, I get almost a perfect identical image to my ideal optimized pan-membrane stain. And this was not great, but it was useful because it meant that what I thought was happening. There is residual primary left on the tissue, still bound to AE1, AE3 that's being picked up by CD44 and 45 secondaries. And so just to give you guys another idea of what's happening, is we are washing off this residual fluorescent signal, but then we're only losing the secondary and the primary staying bound. And then this primary stays bound to tissue as we go through our next steps of labeling. And then it gets relabeled again by my fluorescent marker in white by the anti-mouse secondary. So how can I remove the background caused by the anti-mouse secondary? Well, my first thought was to increase the stripping power before adding on the pan-membrane stain. The reason I thought of this is because I went into the literature and I did a quick bunch of literature, and I found that Acquia Biosciences, which is the company where we get all of our reagents from, they have pre-made kits, and one of their kits has a pan-cytokeratin marker in it with the same clone that we're using. And they said that for their kit, specifically for that clone, you had to use 2 rounds of epitope retrieval, something you did not need for the other markers, which then basically confirmed my assumption that this clone is incredibly sticky and will stick to its epitope better than the other antibodies that I usually work with. And so I did an experiment. So my next step to deal with the background was to run 2 rounds of epitope retrieval and really try and get rid of that primary antibody. And it did not work. In fact, it looks the exact same. It failed. So that failed. And I moved on to my next step, which was to dilute the anti-mouse secondary antibody. And the reason I thought to dilute this is because diluting down the primary at the beginning of my project worked so well. And so I diluted my mouse secondary from 25% to 5%. And while it's not great, it did do something. If we look specifically we'll find that there is a higher contrast of membrane staining around the cells to the background compared to the other image. It's still not perfect. I want to be better. And so I did, so this worked somewhat, and I moved on to another option that I thought up with my lab. And what we decided to do here is to use an antibody FAB fragment before adding on the next, after using an antibody FAB fragment after the 8183, after stripping, adding it on, and then adding on top of that, my PanMembrane stain. And the point of the FAB fragment was to block any residual FC receptors within the tissue to hopefully block any binding of the anti-mouse secondary. And what I found with that is that it did the exact same thing as diluting down the secondary. I basically had the same image twice. We look at that here. These two did the exact same thing. So this isn't bad. It means we're moving towards something. And my next step would be to combine these 2 things together to see if they can work together to remove even more background. But I had one last thing I could work on, and that was switching the staining order. Perhaps I stain AE1, AE3 after my PanMembrane stain so that I don't have to worry about that residual antibody being left over. And that way AE1, AE3 goes into the 8th position. We don't have to worry about stripping it off. It's the last thing that goes on. So I did that. I switched the 2. And what I found was not ideal. So if we look at these images one by one, what you're looking at over here is what's happening in position 7. Everything in orange is the pan-cytokeratin, and everything in white is my PanMembrane stain. When the PanMembrane stain is in position 8, we see that there is overlapping signal between AE1, AE3, and PanMembrane as expected. When the PanMembrane is in position 7, however, we lose a lot of signal, but we're able to maintain the AE1, AE3 when it's in position 8 instead of position 7. That's a good thing. It means we can move AE1, AE3 around. That was exciting. However, this evident loss of signal did concern me because it's not what I expected. Once again, where is that signal going? It looks really good when I only stain with my 1 marker alone on my tissue. So I went again, as I did previously in my experiment, and I flipped through all the other channels within the multiplex that I had stained on this tissue. And I found that there was some cells that were outlined by other markers. If you look specifically in the middle here, at this one here, we'll find if I flip back, it disappears. So same thing that I showed at the beginning of this presentation where I was missing cells in the bottom corner, we're still missing cells even with the PanMembrane stain on it. That was concerning. However, I quickly realized if I layer on PD1 and then also PDL1, to catch all the tumor cells over here. And then, because I'm used to looking at it in white, false coloring it to white, I largely recover my PanMembrane stain, and I'm able to get crisper outlining of all the cells with a higher contrast than I was getting with my other solutions prior to this. So that worked pretty well and was my best solution so far. However, I have my third aim. My third aim was to identify better segmentation software than the current platform that we were using. Now, the current platform that we were using was, I'm going to backtrack a bit there. Cell segmentation can be done in 2 ways. There is nuclear dependent cell segmentation and there's nuclear independent cell segmentation. And what this means is that for nuclear dependent cell segmentation, the algorithm will first go in and highlight all of the blue DAPI signal to label that as an individual cell. And then from there, you will tell the algorithm what your marker is, at what intensity you want to recognize that as a true membrane signal, and then it will search from the middle and expand out looking for that signal. And sometimes it fails, it does not quite get that far, or it goes too far and it surpasses the threshold that you set, and that can be quite frustrating. Versus with nuclear independent segmentation, what we do with this type of software is we give it an RBG image, we give it an RBG image, and in the image I have assigned red to be my membrane markers. So anything that is white here will be turned red and then input into the system. Anything that is blue will be put into the system and labeled as the nucleus. And then the green would be cytoplasm, but I don't really work with that. And the way that this one works is it does not look at the nucleus at all. It just traces the strongest signal from that red channel around where the membranes are. And this works significantly better. If we look at this image here, we'll see over here that the lymphocytes are being accurately outlined, as well as our larger tumor cells in here. They're even getting cells here that did not have a nucleus in them, which is helpful because sometimes you miss the nucleus in our plane section. Versus over here, this is mess. Everything is missing an outline. This is one massive cell. It's not working. So I was really happy when I found Mesmer, because I was able to then upload my new PanMembrane stain with the addition of PD1 and PDL1 into the image, false color it to red, and then look at the results. And what I found is it did work very well. We were able to get almost all the cells accurately. And I would say that next, all I have to do is just tweak it a bit to really make sure we get to the edges. But I was excited about that. Okay. So overall, I was able to achieve all three aims of my project. I was able to improve the input segmentation by removing the background staining for the individual cocktail. I was then able to validate the new stain performance and multiplex images and then troubleshoot that to get to the best that I could. And then last but not least, I found a better segmentation software to segment our images with. And so my next steps will be to leverage AI and other imaging processing softwares to enhance the contrast of the membrane signal from all the other membranous markers, such as PD1 and PDL1, because when we mush them all together, we get slightly more background than just in one channel. But then also just to bring out that really strong, crisp membrane signal that we have, even in light of the minor background staining. And then, so what have I learned? So what I learned over the course of the last year and a half is that when working with large data sets and high throughput analysis, small, easy to miss errors quickly amplify, particularly if early in the pipeline. So in this case, that would be my over segmentation or my under segmentation. Also, in addition, when working in multiplex, the assumption I had when I started in the lab was that when you put all 9 markers together, it should just work. There really shouldn't be anything else going on, but that was not the case. And nor will it be, because anything that goes on before your marker will affect what goes on. But anything that goes on before will affect what goes on next. And that was a big learning for me. Next, I was able to leverage both wet lab and computational skills to optimize cell segmentation. And last but not least, I learned that all antibodies do not act the same in multiplex immunofluorescence. And then I also was very overwhelmed by our technology. While this looks incredibly simple and like 4 key things, each one acts differently. Each one has its own little thing going on with it. And so I quickly learned that. I learned that the order of the markers that we're using within our panel has a big impact on how the image is going to look. I learned that the species of primaries is a big deal because we want to get the most accurate signal, and which species you use matters. I learned that the primary markers could be stickier than others. I also learned about spectral bleed through. I learned that we can optimize each concentration you see here. We can optimize the concentration of the primary antibody, the secondary antibody, as well as we can change which type of polymer we're using. We have 3 different types we can pull from, and I can change the concentration that we use of the fluorescent marker as well. I also learned that all cases are heterogeneous, which means that sometimes one patient will have a phenotype and the other patient won't. And if I'm not looking at enough cases when I'm optimizing my panels, I might miss that phenotype, which might mean that I might be missing staining. So I have to make sure I look at many different cases as well as many different cancer types when optimizing my stain. And last but not least, finding the right software is just as important as using the best image. And that's everything. Thanks, guys.
Questions with the, EVs?
Yeah.
That was a great talk and I learned a lot and I hear you made a lot of useful progress. So I can tell that in a qualitative way and also wearing my editor's hat. I would anticipate that a reviewer would say, well, how do you prove that it's an improvement? What kind of quantitative measures would you bring to bear to show that this works better than what you were using before, or what the standard in the field is, and what are your thoughts about that?
We have, and that kind of is our next step. This morning we were talking about it and we're hoping to send the images down to Johns Hopkins and use their AstroPath pipeline, which is the current best way to segment our cells, and just look to see if the previous images that they have with the old PanMembrane stain on it, how those are segmenting versus how my images will be segmented when put through the same software. And then compare the two. But quantitatively, I think we would do the same thing that I saw with the Johns tablet earlier in our presentation, and just do cell counts and look and see if there's any difference there, as well as look at the phenotypes after phenotyping is done and see if there's a major difference.
So manual cell counts versus automated?
It would be the cell counts based on what the image analysis software would count, because it counts cells.
How do you know which one's better?
I think we would go in and look closely at the images, much like we did previously, and see if the tumor cells are over-segmented or under-segmented, and show that. I'm sorry.
No, I think you're probably gonna need some manual or some gold standard to compare this to, both methods to. I think it would work great and would be a nice project.
I'm curious about using, go ahead.
A quick question. So, when you're segmenting the cells that don't have any of the other markers, why is it important to segment those cells that don't have any of the phenotypes that you're looking for? Like I'm seeing about the one image where there's an area of cells without any markers. Why is it important to even know that those cells are existing down there?
That's a good point. Currently, they do get grouped into the other category when we're working with them. But I think the key point is that we just want to make sure we're getting every single cell. So we want to make sure that when we're looking at the tumor microenvironment, we're looking at it accurately. We're properly understanding how the different clusters are working together, what the cell densities are within those clusters. If we aren't getting that correct, we want to, because we're missing other cells, we'll get the densities incorrect, and from there the data will go. So you just want to make sure that we're getting every cell so that the numbers that we pull from the images are as accurate as possible for what we're looking at.
I'm very curious about using CD64 as a marker. 44. So CD45 makes sense. My understanding is any hematopoietic cells are expressing. And in the case of 44, though, you'd actually expect there to be more expression of it on cancer cells than on hematopoietic cells. Is that what you see? You didn't show us examples where you just looked at each of the three of them individually. So, but you've probably seen that in your data by now. What do you see in that regard?
I have not largely worked with them independently in a long time. They've been in a cocktail for quite a while. When we did dropout experiments earlier on in my master's, we found we didn't really notice it on the tumor cells because ATPase was there. It's
I thought I could notice it in the slides that you were showing, because it seemed like areas that you had especially when you were looking at that background issue, that was enriched, and I think you pointed that out, that the tumor and cancer cell area, that's where you saw the highest levels of background.
Yes,
it seemed to make sense to me if CD44 is more highly expressed in the cancer cells compared to the immune cells, then you would expect that. Anyway, it's something I'm curious about that because sodium potassium ATPase makes a lot of sense. You indicate why it's thought to be pretty much everywhere. I would imagine that there are other proteins that would fall in that category that people have tried. not to mention lipid stains that people have tried.
Yes.
So how you settle on, and CD44, 45 makes sense in the immune cells, but I'm just not sure how many other potential antibodies there would be. And when it comes to that, you had a bit in your conclusions about not all antibodies being the same way, and of course that's a problem that we've been dealing with for decades, using antibodies that they all behave differently, and so that's something you have to work through, antibody to antibody on this. But I'm curious, when you're doing this, trying to set up a protocol that you hope will then be something that can be used widely, what about batch to batch variations and what you think is the same antibody from the same supplier? Have you ever looked at that? Or had problems with
I
that?
have not looked at that personally. No, I've not had problems with that.
Is that something that happens?
Yeah.
There's probably several people in the room that can attest to that, with the types of experiments that they're doing. And whether it's because of thought on the loading dock, you didn't know, or some other issue, you never know.
We do keep an eye on it. And I have found that with working with my images, I do pick up on changes quite quickly that are not there. For instance, when I was having blocking issues, I did notice that almost immediately in my images after working with them in singleplex for so long. So with that, while we might have batch to batch effects, we are careful with what we order. We do try to make sure everything is the same. And then we do go, like, we have seen issues in the past where there have been things are not consistent, and we go and look at them when they're caught.
Because I don't know what happens in clinical labs, but I assume that there's QT, quality control analysis that's done, probably even from batch to batch.
When we start seeing clinical specimens, we will include serial sections of a TMA in every staining batch. So part of the astropath image processing is actually to normalize the fluorescence signals across the batches using those TMA sections. So to account for not only antibody variation, but also the fluorophores and like all the other, yeah. It is important because we do want to measure the intensity in a meaningful way. So it is one of the things that.
I want to dominate a question. I have lots more questions, but other people, hand over there. And so while you're thinking about your question, another question I have is about you found a better software. And whenever I hear that sort of thing, people switch. I wonder, okay, what's different about it? Because I think a lot of people my age anyway, maybe not so much the younger people that know how to write in R and that sort of thing. But to me, it's black box. Do you have any idea what was different about the software? Why it was better?
The black box in my case was actually Inform, which was the one that I started with. That was the software I initially started with. I was having a really hard time understanding how that one worked and how it was optimized and why it was working well. Versus with Mesmer, what I ended up finding is they had a very good open source paper, and then they have very good open source code, and I can download the code and go through it myself and see how it's working, which was really, really helpful. And I did that, and I can also place it into other code that we're working with as a function and adjust it accordingly to have it get all the borders that I needed to get. The original software I was working with was isolated. I couldn't play with how it was working. I had to order the different antibodies that were membrane antibodies in a hierarchy, which didn't make sense to me because if it shows one was better than the other, it would often look for the first one and then totally miss the second one that I listed. And that was really frustrating. So with Mesmer, I said it was a better software. It was also, when in its development, it was also kind of somewhat open source, in which they had a lot of people go through and make sure that each image was correct. They would send it back to the AI and then train it again. And they went through iterative loops of that to create this sort of ground truth. And then from there, they were able to show that it worked across different tumor types, tissues, and with different markers. Because if I can upload any RBG image and just make sure that all of my membranes are in the red channel, it will go and it will outline all the dark red. And it does a good job in comparison to what I was working with before. Yeah.
So in that regard, you tried one with nuclear stain and without. And so I was optimistic when I heard you say we were going to do this. Oh, that makes sense. using more information, it should be working better, but were you,
No.
so what do you think is going on with that? Wouldn't you expect more information about where you can see a nucleus to actually improve that?
Yes, yes, and no, because a cell is larger than the sections that we take from our histological samples. We sometimes miss the nucleus in some of our images, so it's not there, and then we miss that cell. And that's not great when we're trying to make sure that we get every single cell labeled accurately within the tumor immune microenvironment.
Yeah, I guess you can see that. Some cell types, that's gonna be more of a problem.
Like with tumor cells.
Wouldn't be a problem with T cells, but it would be.
Gabby,
Yeah.
Great presentation.
thank you.
I did have a question about, I guess, the logistics of the PanMembrane. When you were describing the three components of it, I was wondering, is there a difference between adding those together as opposed to having them stained separately? Is there a caveat to having them all in one sort of stain?
We stain them all at once because we want to label them all with the same color. And then at the same time, we only have so much space within our panel. We only have 9 markers. We're not going to use 3 of them to outline the membrane, but it does a good job all together at once. Putting them on one at a time on the tissue, our automated stainer does a good job, and we have not found the cocktail to have any issues with putting them all together at once and then labeling it as such.
Any other,
Thank you for the wonderful presentation. I was just wondering, the software that we use for the analysis, does it take into account the signal noise removal?
Because back in time, back paper. I'm sure it does. I don't remember exactly. I can always send you the paper if you're interested to see if it does.
I believe another thing to look at apart from enhancement is to look at the noise.
Mhm.
With that, you might get like a clear view of what you're looking at. So noise removal most times takes care of the issues that you have with your images, noise removal and enhancements.
Yeah, I think that is hopefully the next step for what I'm gonna do, is just work with the images themselves and move on from the wet lab stuff. Thank you.
It's a great talk, beautiful images. You know, journal editors love these sorts of images on the front covers, so you shouldn't have trouble selling this. And if the science phase fails, you can sell it to some people who are making fabrics or something. Seriously, epitope retrieval.
Mhm.
You didn't talk a lot about that, and that's a real problem because you've got some epitopes that are going to be extracellular and some which are going to be intracellular. I mean, they're all membrane related. So how did you come to the right conclusion about epitope retrieval?
We have about 2 different ways to go about epitope retrieval, 4 different ways to go about epitope retrieval in our lab, and we test each one when we're looking at each marker individually to see which one works best for that marker. I've never really thought of it from an intracellular to extracellular standpoint before. Because when we slice our images, I just kind of think of them as just being on the surface and that the intracellular and extracellular are exposed the same. They're not like full spheres.
I think one of your pancellular markers had had intracellular components for sure. I think the epitope was intracellular.
It was, but when it's sliced in half, it's all exposed to the same amount of heat and the same amount of acid or the same amount of solution as well. So it's not trying to get through the membrane, it just sits on top and then it just gets all the proteins as such.
One other quick question.
Yeah.
Therapeutically, people are beginning to use things called nanobodies now. So camelids, as you may know, so camels, alpacas and so on, don't make antibodies which are very big, 150 kilodaltons. They make variable chain, heavy chain antibodies, which are about 15 kilodaltons in size. They otherwise look exactly like antibodies in the way they act. Is anyone using nanobodies for imaging?
Not to my knowledge.
I mean, the one thing they can do, because they're so much smaller, is that they can see epitopes that regular antibodies cannot see. So it might be, I don't know whether there would be disadvantages, but for imaging studies, it might be something that would be useful to better define things you want to look at. So you might search nanobodies and imaging.
That would be interesting. Yeah.
I just wouldn't want to bleed in that camel.
They spit a lot. What are you gonna do this way? You can make just stand a long way away.
So water, they need less water.
I don't see any more hands in the audience, no more wisecracks, nothing, you know? Okay, then